Recording's in progress.
All right. All right. Good morning, everybody. Welcome to the research seminar for direct measurements of ozone sensitivity to NOx and volatile organic compounds in the South Coast Air Basin. My name is Shang Liu. I'm from Research Division of California Air Resources Board, or CARB. I'm the manager of this contract. So this project is funded by CARB, and the PI is Professor Michael Kleeman from UC Davis. This is the agenda of today's seminar. I'll start by giving you a background introduction, and then the project team will present findings from the project. After that, there will be time for questions and comments, and the seminar will end by noon today. And before starting the background introduction, there are some housekeeping items that I want to describe. So the meeting will be recorded. The recorded video will be posted to the CARB's contract webpage, and here's the address of the webpage. So I'm going to copy and paste this webpage to the chat shortly. And if you have any questions or comments, please type them in the chat or raise your hand during the Q&A session. So this project focuses on ozone, which is a criteria pollutant. So ozone has adverse effects on public health and the environment. And ozone is not directly emitted from sources. It is produced chemically from reactions of NOx and volatile organic compounds, or VOCs, in the presence of sunlight. And because of its adverse effects on health and the environment, CARB and California have been making continuous efforts to reduce ozone levels. And because of these efforts, ozone air quality has improved during the past decades. However, now we are facing a challenge. We are experiencing a slowdown of ozone reduction during the past decade. And here I'm showing you an example for the South Coast Air Basin. The y-axis is the ozone design value, and the x-axis is the year. And we can see that during the past 10 years, there's no change of ambient ozone design value. And this is just an example for South Coast Air Basin, and we see this feature in many other regions in California. And as a result, California still has the worst ozone air quality in the United States. And in particular, the South Coast Air Basin and San Joaquin Valley are classified as extreme non-attainment areas. So we don't know why there's a plateauing of the ambient ozone level in California. And this may be because of the nonlinear response of ozone to its precursor volatile organic compounds and NOx. Here I'm showing you how complex this nonlinear response is. This is the so-called ozone isopleth. It's the ozone concentration as a function of its precursor NOx and VOC concentrations. So there are two ozone formation regimes. So one of them is the VOC limited regime, meaning that in this regime, if we reduce VOC concentrations, we can reduce ozone concentration in the air. But if a region is in a VOC limited regime and we reduce NOx, we may experience an increase of ozone concentration. And another regime is a NOx limited regime. And in this regime, if we reduce NOx concentration, we can effectively reduce ozone concentrations. So we really need to understand where we are in terms of the ozone formation regimes. And how do we know that? Typically, most studies rely on models or indicator measurements, such as the satellite measurements of the ratio of formaldehyde to NO2, or comparing the measurements during the weekdays and weekends. But there are very limited direct measurements, and these measurements are really needed to justify the modeling studies or the indicator measurements. And this is exactly the purpose of this project. So these are the objectives of the project. Task one is to configure mobile ozone chambers that can be deployed in the field for field measurements. And task two is to verify the proper operation of the chamber system, making sure it's working fine. And task three is to make primary measurements of ozone sensitivity in the South Coast Air Basin. And the last task is to perform statistical analysis and hypothesis testing of the results from task three. So I wanted to mention that task three, the primary measurements were planned for the summer of 2020, but because of COVID, the measurements were delayed by a year. So the team measured ozone sensitivity in the South Coast Air Basin in the summer of 2021. However, because of this COVID, the team found an alternative. They did a measurement in Sacramento for almost a year during the pandemic. So I'm really impressed by the dedication and enthusiasm of the team, and I'd like to take this opportunity to thank the team for their efforts. And I also want to thank the CARB's MOD for providing support for the Sacramento measurements. And to the best of my knowledge, the Sacramento study is the first long-term measurement of ozone sensitivity that captures the seasonal variation of ozone sensitivity, and the results are really exciting. They are already published in atmospheric chemistry and physics, which is a very good journal in atmospheric chemistry field. The delay of the measurements actually gives us an opportunity to combine several field projects together to form a bigger campaign, which is called Recap, a summer field campaign in the summer of 2021. This campaign was in collaboration with other agencies, and as you can see, we have very comprehensive measurements. And in particular, this project is one of the backbone projects of this bigger campaign. And the team conducted measurements at Pasadena and Redlands for a couple months using 2 sets of ozone chambers, and the results are really good. On top of that, the project team also conducted some modeling studies, which are complementary to the chamber studies, and provide some suggestions for future ozone control strategies. So I'm going to stop here and hand it over to Mike for the details of the results.
Thanks, Sean. I'll go ahead and share my screen then. So, all right. Can you confirm that you can see the presentation?
Yes. Just go ahead.
All right. Well, thanks for that introduction. As Sean said, this was a project that kind of grew in scope as we went along, partly because of COVID, and then partly also because we were just genuinely excited about the results. I think that the study yielded some very interesting trends, and it was exciting to work on it. And I think that, I'm very proud of how this all turned out. The whole team, I think, did a great job, both at the university and then all of our partners at the various places. And I'll go through and try to acknowledge everyone at the end of the presentation. But I want to especially acknowledge my PhD student, Shenglun Wu, at the beginning, since this was his dissertation work and he was involved in all aspects of what I'll present today. And so this is really his PhD thesis that we're presenting today. So as Sean introduced, we have this question about how to make continued progress on reducing ozone concentrations in California. We've made good progress over the past decades, 5 or 6 decades, but we're now hitting sort of a decade in which their progress has not been as rapid, and we may even see slight indications of increasing ozone concentrations. And we have, of course, all the techniques that Sean mentioned, model predictions and, weekend, weekday trends, observations with satellites, but really some in situ measurements directly probing this would help add to the weight of science and to build confidence that we fully understand what's going on and we can explain it. Is it really just a case that we are, in the VOC limited or the NOx rich part of the isopleth, and we have therefore NOx controls are going to increase ozone or have little effect on ozone? Or is it a case that perhaps we're targeting the wrong sources in our emissions control program? And so we wanted to design measurements in the field that could add to the weight of science to help us be more confident in the answers to those important questions. So what I'll talk about today then is just a really quick review of that ozone chemical regime diagram that Xiong introduced. And then we'll describe our system for making those in situ measurements, ground-based measurements of the O3 sensitivity. I'm going to describe then the results of that first field campaign that happened during COVID, during Sacramento 2020. And then we'll compare that to satellite measurements to try to get a broader perspective over all of California. And then we'll discuss the expansion of the measurement system to 2 trailers instead of just 1, and then its deployment in the field campaign as a part of the recap field campaign, and then integration with VOC measurements, again, supported by partner agencies, and then the modeling analysis and the overall conclusions that we'll draw. So, this was really a team effort that used a lot of different techniques. There were a lot of people involved in it. And as I said, I'll try to acknowledge everybody at the end. So as Xiong introduced, we have this, isopleth diagram for the ozone formation that tries to capture that nonlinear behavior. And really what it comes down to is competition for the OH radical. If the hydroxyl radical reacts with VOCs, then that is going to produce more radicals and eventually entrain oxygen into the system such that it produces ozone. But if the hydroxyl radical instead reacts with NO2, then it'll chain terminate, it'll produce nitric acid, and it will shut the chemistry down, it will quench it, so to speak. And so this ratio of VOCs to NOx is a really critical parameter that we've known about for decades and decades. What we don't know right now, or what we want to make measurements of, is exactly where are we on that isopleth diagram. If we go out and could make a measurement, what would that find? And so there's some important questions that are tied up in that. Do we just need to recognize that we're climbing over the ridgeline in the isopleth diagram right now for the past decade or so, and we just have to continue on that process until our NOx emissions are reduced sufficiently to get on the other side of the ridgeline where we're NOx limited? And then we'll see, further progress in reducing ozone concentrations. In the context of that, during COVID-19, we had a tremendous reduction in on-road vehicles and traffic. And why didn't we see ozone concentrations decrease during that time period? And so that, I think, also adds to the question swirling around, what we're trying to answer in this project. And then, of course, there's been a realization that as motor vehicles have been cleaned up and other sources have been cleaned up in recent years, maybe we need to focus on some of the, other sources that haven't been leading candidates for VOCs in the past. But maybe if we control those other major sources, things like volatile chemical products, personal care products and so on, could be part of the remaining sort of reactivity of the atmosphere. And so we want to try to, address all 3 of these questions in this study, where we are going to try to use in situ measurements and modeling to better understand what's going on. So the first field study, as Xiong introduced, happened just as COVID broke. We were gearing up to go to Southern California, but instead, we basically configured everything and deployed it in Sacramento for a year as a way to do something productive during the COVID pandemic. This was a natural experiment, of course. We took a lot of vehicles off the road during that time and greatly changed our emissions. And so in addition to making a year of measurements, we also were able to understand how those measurements fit into the bigger picture of greatly reduced vehicle traffic, at least for the first several months of the pandemic, and help understand then what the traffic contribution to the ozone formation might be. So again, with partners at CARB, we were able to deploy the trailer that contains the measurements in Sacramento and power it and run it for an entire year. We could work out some of the kinks in the system as well, which made the ultimate field deployment in Los Angeles a little bit smoother. So we all know what downtown Sacramento looks like, but we have major freeways nearby, we have rail stations, we have commercial, and residential neighborhoods all around us. And so, it's a mid-sized city in California, would be the biggest city in many states if you transplanted it to many states. So there's, it's a lot to learn here in Sacramento. So what does our system look like? Essentially, we have three 1 cubic meter smog chambers that we take to the field, and they are all loaded with air from the ambient. One of the chambers is not perturbed at all, and so it would be our base case measurement. One of the chambers is going to have NO2 added to it, a NOx perturbation added as NO2, and then the other chamber is going to have a VOC mixture added to it. That is based on Bill Carter's sort of surrogate for the urban atmosphere in California. So this is the mini surrogate, the sort of minimum mixture that would capture some of that reactivity. And this is also the basis for the ozone reactivity scales that we use to evaluate different VOCs. And so we compare those two perturbed chambers to the base case chamber during a 3-hour aging experiment to try to directly observe how the ozone response changes. If we're in a VOC limited or a NOx rich regime, when we add NO2, we expect to see ozone decrease. And if we are in a NOx limited regime, when we add NO2, we expect to see ozone increase in that. Now, this was not, I should say it positively, this was designed to be as atmospherically relevant as possible. And so again, we're trying to add small amounts of the NOx and the VOC, and we are trying to drive the system with photochemistry that's representative of a typical day in California during the summer. And so all of this is geared to be as atmospherically relevant as possible, but it is under controlled conditions. And so we use black lights, not natural sunlight, and we are trying to reproducibly do this experiment every day and just watch how the trends evolve over the seasons, essentially, and as a function of location eventually when we deployed it in Southern California as well. So our monitors were geared towards the ozone formation. So we had an ozone monitor, of course, and a NOx monitor. We also had an NOI monitor, and then temperature and relative humidity monitors in the first application. We did have an AMS at times that was monitoring in the chamber. I won't present those results today. And we did at times take sort of more detailed VOC measurements in the chamber after the reactions occurred. But again, I won't present those today. So we have a little bit more detail sort of that's still being analyzed. And I won't go into all of that, but I will focus on the ozone results. And that's complicated enough in an hour-long seminar. So hopefully we'll get through that and have everyone understand what we did. We filled the chambers between 10 and 12 every morning. And that was done because we wanted to do a 3-hour photochemical exposure after filling. And we knew that the ozone peaks sometime between 1 and 3 in the afternoon. And so the timing just suggested that was the right time to fill the chambers. In an earlier sort of pre-study of this, we did do experiments at nighttime where we would fill the chambers at night. And what you reliably see, and when you do that, is that the system is always NOx rich at night because there's no mixing at night. You still have NOx emissions. And a lot of the VOC sources, like the biogenic emissions, will shut down or be minimized at night. And so, we thought we wanted to try to pick a fill time that was most representative of the photochemical cycle. We certainly could fill at different times and look at how that evolves. But again, our focus in this study was peak ozone. And so that sort of drove our, fill times and our exposure times. Now we mixed the perturbation gases into the chambers halfway through the filling, and then we continue filling as a way to mix everything up. There is no sort of stirring of the chamber during the experiment. although there will be some sort of thermal mixing of because the lights do generate some heat, and so you will get some thermal mixing of the chamber, but there's no mechanical agitation with a stir bar or anything like that. We begin each experiment then with dark measurements before we turn the lights on, and then the lights are turned on again at that sort of atmospherically relevant UV level for 3 hours. And we measure the ozone and the NOx and the NOI concentrations throughout that 3-hour period. Now, I think the strength of the experiment is that there's one instrument package that just rotates between the chambers using an automated valve system. This is all controlled by LabVIEW. And so the instrument package is pretty stable, and so we don't have to worry about biases between the instruments. But what that does mean is that we get a 10-minute snapshot of measurements in chamber 1, followed by 10 minutes in chamber 2, followed by 10 minutes in chamber 3. They're offset in time, and so they're not directly comparable. And so in order to sort of project the ozone concentrations at the end of the 3-hour experiment, we would fit a line through the measurements. And typically it was very linear. And so we have high confidence that that sort of method of projecting the final ozone concentrations so that we can compare all of them at 3 hours, not 10 minutes or 20 minutes offset from each other, is a fair comparison. And so that was part of the analysis as well. Now, again, just to try to convince people that the measurements are atmospherically relevant, we compared the ambient measurements outside the chambers to the measurements inside the chambers. So the measurements inside the chambers in each of these, I've got 9 months worth of data shown here from April through December, are shown as black dots. And these are typical weeks in each of the months. So the black dots, start out at the same concentration as the ambient measurements, the lines in each of these plots. In many cases, you'll see that the slope, the sort of rate of ozone increase inside the chamber matches pretty strongly with the rate of ambient ozone increase. But you'll see that the lines sort of peter out and sort of drop off. And that's because the real atmosphere is mixing. So in addition to the chemical production of ozone, you're getting the mixed layer sort of increasing over time, and that's diluting the ozone concentrations in the ambient. But of course, the chamber doesn't measure that. So I guess one important realization of this is that our experiment is focused on the chemical production of ozone, and it doesn't account for transport and mixing and those other things. We think that the chemical production is one of the most important, if not the most important features when we're thinking about whether we're NOx limited or VOC limited. And so we think that this is an important piece of the puzzle, but we want to acknowledge the complexity of the real atmosphere. And, if we can, use our methods to gain confidence in the models, it really is the models that incorporate all of this together simultaneously. So we still think that chemical transport models are a really important part of ozone design programs, but we're trying to provide corroborating evidence or add to the weight of evidence to convince ourselves that those models are right. Okay, so we're also going to try to use Tropomi satellite measurements to try to verify that our measurements are correct, and that understand how point measurements at one location sort of what they mean in the broader context across California. So the Tropomi is not a ground-based measurement. It's a column measurement, and it can only see certain things. It can see formaldehyde and NO2 really well. But we think that those are useful and critical in sort of interpreting our ground-based measurements at one or two locations, understanding how they compare to other locations, and then also trying again to build a weight of evidence approach in our interpretation of our data to make sure that we've got our story straight. All right, so just some trends then in Sacramento on the right here. You can see on the upper plot, then I've got month on the lower axis going from February out to December. And then we've got the measurements of the NO2 in the chamber and sort of a metric of the VOC, and I'll go into that in a second. So in Sacramento, and I'm presenting all of this in the monthly readings as box and whisker plots, because again, the atmosphere is a sort of a messy place. And so you have to do some statistics to really interpret the trends here. And so what we see with the measurements, the box and whisker plots, is this increasing trend of the NO2 concentrations sort of as we go throughout the year towards the end of the year. We've got those concentrations increasing, and that reflects both the use of fuels for heating as we get into the colder months, and then also changes in atmospheric mixing as we get into the colder months, sort of at this site. Now, I want to point out that we do have weekday differences. And so if you look at, well, I guess we've got chamber and chamber no fire. So we have wildfire-driven differences in this plot. And we do see some differences on days when we had wildfires in the late summer and early fall. I also want to point out that the Tropomi measurements sort of yield the same seasonal trends when we're looking at the column averages. So the Tropomi measurements are shown as diamonds in this. Of course, that's, again, a column average. It's not a measurement at the ground level. So it's on a different scale. You can read the scale on the right-hand side of the plot. Instead of ppb, it's a column average. But again, that spatial trend is matching what we saw in the chambers. If we look at the measures of VOC, we see sort of an increase in the summer months and then a decrease later on. And at the time that we made these measurements, we were using sort of a combined metric of the carbon monoxide concentrations multiplied by a factor to account for the emissions of biogenics sort of responding to temperature. We also think that could probably capture the same trend as sort of evaporative emissions from gasoline vehicles, sort of in retrospect. But we see this trend of higher concentrations in the summertime. Now, again, we wanted to build confidence in that metric was meaningful. So we compared to the Tropomi formaldehyde measurements, the column measurements, and the diamonds in this plot. And you see the exact same trend, different units, of course, for a column measurement versus a ground-based measurement. But you see those exact same sort of seasonal trends there. So we do believe that this CO biogenic sort of combined metric that we're using to sort of quantify the changes in the VOC concentrations is meaningful. We also then, because the analysis sort of took place over many months and even a year after we made the measurements, some of the isoprene measurements from the PAM sites became available, I think. And so we were able to compare it directly to those isoprene measurements as well. And so, again, all of those things sort of are consistent, and we see this sort of increase in the summertime VOC measurements. So that's really important, all of that background, to understand then our sensitivity measurements. These are the first measurements that I'll talk about for the ozone sensitivity. And we're presenting it again as box and whisker plots as a function of month. The upper panel then shows, on the left-hand side, shows the response of ozone to a NOx addition, that NO2 addition. And the dashed line sort of going through zero in this plot would be no response of ozone. Anything above the dashed line means that ozone increased when we added NO2, and anything below the dashed line means that ozone decreased when we added NO2. So you're directly measuring whether you're NOx rich or NOx limited through this diagram. And what we see is that there's a seasonal trend, that in the cooler months, we're NOx rich, where adding NO2 decreases ozone concentrations. And in the hotter months, we're NOx limited, where adding NO2 is going to increase ozone concentrations. Now, this is corroborated again by the Tropomi measurements. The Tropomi measurements in the diamonds here, we can look at the formaldehyde to NO2 ratio, and we see again that same seasonal trend. And we've drawn the line, the dashed line on the Tropomi scale at about where we think the break point would be between that ridge line between NOx rich and NOx limited conditions. And so the Tropomi measurements are in really good agreement with the ground-based chamber measurements, and we think that this all makes sense. If we look at the lower plot on the left-hand side, where the response of ozone to adding a VOC, we always see ozone increase slightly when we add a VOC. Those lines on the isopleth don't really bend backwards very strongly at all. And so we see sort of the mirror image of the NOx perturbation as we would expect. And so again, this corroborates and sort of it adds to the weight of evidence that our interpretation of these chamber results is correct. We see the VOC response sort of mirroring the NOx response. And so it all sort of seems logical. And again, the Tropomi is in quite good agreement with the VOC response as well. Now, if we plot this instead of by month, if we try to look at the chamber response as a function of the ambient ozone concentrations, what we see is that in Sacramento, at least at the time that we made these measurements, the lower ozone concentrations are in a NOx-rich regime. The higher ozone concentrations seem to be in more of a NOx-limited regime, at least in Sacramento. And so that helps us sort of think about the design problem for ozone. But again, we want to be able to have a broader perspective of that. So we're going to draw in the Tropomi satellite data to try to spread our vision a little bit and look across California. Now, we can also see the weekday trends in the chamber measurements. So, you can look on the lower plots for the NOx perturbation and the VOC perturbation. Here now, the sort of solid box and whisker plots are the weekdays, and the open box and whisker plots are the weekends. And you always see more NOx limited behavior on the weekends. And so again, this corroborates our interpretation of the data. It confirms what we think we already know about weekend versus weekday trends. And so it's all consistent, and it builds confidence in our interpretation. Now, we wanted to also think about these seasonal trends and try to build an isopleth plot based on measurements. Those isopleth plots that we look at oftentimes are built on model calculations, and we wanted to be able to try to make a measurement of the isopleth plot. And so what I'm showing in this plot is the VOC sort of metric that we use on the lower axis, the NOx metric on the vertical axis, and I'm showing the ozone concentrations at the end of our 3-hour experiment over this year of measurements as the color of the dot that you can see. And so you see the red, the hotter colors corresponding to the higher ozone concentrations, of course, occur in sort of as you move from the lower left to the upper right. Now, the arrows are the perturbation results. They're showing how the ozone concentration responded to either a NOx perturbation or a VOC perturbation, sort of combined into one arrow. The direction of the arrow should point at the ridge line of the isopleth. And to a large degree, it does. And so you can see then that, we can form that measured isopleth diagram from our data set. And you can see how the seasons change. The blue circles here corresponding to different months of the year show how we move around seasonally on that isopleth diagram as the ambient concentrations of NOx and VOC are changing. So a lot of power in those ground-based measurements. Now, using the Tropomi data, we wanted to broaden our perspective away from just Sacramento and one site. What does it mean across California? And so we looked, it was an integrated team effort here with researchers at CARB and UCD trying to interpret the Tropomi data and to provide that perspective. So in this plot, I'm showing the formaldehyde to NOx ratio, which again, we think is a key indicator of where we are on the isopleth diagram. Anytime we're in the green sort of colors, we think that we're NOx rich. So adding NO2 would decrease the ozone concentration. And anytime we're in the red color, we think that we're NOx poor. And so adding NO2 would increase ozone concentrations. And you can see the seasonal shift across all of California. The Sacramento site is measured as the small triangle in the center of one of these maps. It's difficult to see, I guess, but you can see that what happened in Sacramento was kind of mirrored across the entire region to different degrees. Certainly, we have consistent NOx-rich conditions in our major urban cities. So Los Angeles is always NOx-rich throughout the entire year. San Francisco, sort of similar trends. But you can see the more rural areas then go through a much stronger seasonal cycle. And so we think that this provides perspective. Does one size fit all in terms of a control program for California? And that's a legitimate question to ask. So we can try to, understand where exactly that split between NOx-rich and NOx-poor conditions happens in the Tropomi data by comparing to our chamber measurements. And we come up with a ratio of around 4.6 for formaldehyde to NO2, which we think is in the range of sort of values that have been put together by other researchers looking at this data set and perhaps looking at weekend and weekday trends and things like that. So we think that again, this builds a weight of evidence that we're interpreting our measurements correctly. And then it also allows us then to analyze different air basins and, see which ones as a function of month of year sort of cross into that NOx rich regime or NOx poor regime in the summertime where NOx controls perhaps could reduce ozone concentrations. Again, it's hard to capture the complexity of that spatial plot that I had on the previous diagram with one line for the South Coast Air Basin. The outlying regions of the South Coast Air Basin may well go into the NOx poor region during the summertime, but the core downtown LA area definitely does not. And again, that will come out in the ground-based measurements that we show for recap in just a little while. So we'll see that again is going to be matched. So this again is just another plot as a function of month of year, looking at where we still think that we are sort of NOx rich in the darker shaded areas versus NOx poor in the lighter shaded areas. And what you see again is that trend where in the outlying areas, you have more of a seasonal cycle where you can cycle between NOx rich and not poor depending on the month of the year. But in the inland or in the urban core regions, you're staying pretty consistently NOx rich throughout the entire year. And again, we think that will be, this is all based on tropomi, but we think that our recap measurements where we had two sites at different sort of distance downwind of the urban LA will corroborate this basically. So we'll show that in a second. All right, now a lot of great comments came up during the review of this study, and we built a model then to address some of them as a practical way. What was the effect of the temperature in the chambers being a little bit warmer than ambient? What was the effect of using UV lights in the chamber instead of natural radiation? What was the effect of having a 8 ppb NO2 perturbation? What if you had a 2 ppb NO2 perturbation? Maybe you're jumping across the ridgeline. A lot of really great sort of comments and challenges, and that's how science works, right? We challenge each other. So we responded to that by building a chamber model to try to help us interpret the measurements. And what they showed is that, our conclusions were solid. Certainly the numbers move around a little bit in response to all those factors that I listed, but it didn't change the overarching conclusions that we reached, which is that you have this seasonal cycle, and it looks like, it's consistent with, sort of a cycle of biogenic emissions is what we interpreted from this. What VOC could be going up strongly in the summertime across California, not just in the urban locations, but across California. And that really points to the role of biogenics in driving that seasonal trend. So our conclusions from all this then, we made these measurements for a whole year in Sacramento, sort of an ad hoc field study in addition to what we had originally planned in the proposal. And then we see this seasonal cycle of the ozone chemistry going from NOx rich or VOC limited in the cooler months to more NOx poor in the hotter months, and then back to NOx rich in the cooler months at the end of the year. And it's consistent with the cycle of biogenic emissions in California. It's happening not just in the major cities, but across California. And we think that that's very consistent with, having the biogenic emissions driving some of this. And so, we were excited then to take it to the next step. So the next step was a field study, the original field study that was planned in Southern California. Again, because of the delay of a year, we were able to coordinate with other things. And so we were there as part of the recap field study, where we had collaborators from NOAA, and then CARB was also able to gain additional instrumentation and bring it to the sites. And so we were able to also leverage other funding. So we were successful in getting funding to build a whole second set of chambers, a second trailer, if you will, so we could deploy 2 field sites at once. And so we went early and we stayed late. The main recap field study was just 1 month. We were in Southern California for 4 months. And we were deployed at 2 sites, at Pasadena and at Redlands. Pasadena was the core site for the measurement campaign. And again, thanks to our hosts at Caltech. And then Redlands is the design site for Southern California. It has the highest ozone concentrations, or at least it did in 2021. And so, again, thanks to our hosts from the South Coast Air Quality Management District for getting us set up at that site. So we've got 2 parallel chambers now. We wanted to, of course, make sure that they gave the same answer. We didn't want any biases introduced just by different, slightly different trailers or chambers. So we made some measurements both before and after the campaign. That was really critical, actually, because we did detect a sort of offset in the NO2 measurements in one of the trailers because of that. That happened during transport to Southern California. So we were able to correct our data. So it was really critical to do those quality control checks before and after. And we had good agreement for both the measured final ozone concentrations, the measured response to NOx perturbations, and the measured response to VOC perturbations. And so again, we think that those 2 trailers are essentially interchangeable. They were not biasing our interpretation of the results at all. Again, just a really quick slide to show across these 4 months that we were making atmospherically relevant concentration measurements. The solid lines, again, are the ambient monitors beside the trailers where the chambers are located. The dots are the measurements inside the trailer, inside the chambers, after we get that grab sample from 10 to 12. Again, we're starting out at the same concentrations. We don't somehow bias the measurements or the sample when we bring it into the chambers. And we've got the same sort of general rate of increase. You see again, though, that we don't have atmospheric mixing or transport in the chambers. And so you see the chambers continue to go up after the real atmosphere sort of maximizes and starts to decrease. And that's because the effects of mixing in the real atmosphere. So we're measuring the chemical production of ozone. And again, we'll rely on the models for the sort of complete picture. There was a lot of great measurements during recap. We made VOC measurements under sponsorship again from CRC, and I'll talk about that shortly. But we had a lot of great real-time measurements from our collaborators at NOAA and CARB, running various instruments out there. And so that really allowed us to dig deeper into the composition of the VOC, maybe do source apportionment calculations for the VOC as well, much more than what we could do in Sacramento during that pre-study. I won't belabor things. We were there from July to October, so the temperature got a little bit sort of went through a seasonal cycle. The relative humidity went through a cycle. The gasoline changed. I'll talk about that in a second as well, because we use summertime gasoline versus sort of winter blend gasoline in California. So lots of things sort of moving around during this 4-month measurement period. Now, the initial NOx concentrations, I'm showing them here as a function of weekend and weekday. We see that weekday pattern that we're familiar with across July, August, September, October. And we see, concentrations in Redlands are a little bit lower than concentrations in Pasadena, again, because of proximity to downtown LA, where all those freeways come together and you've got such a high population density and a lot going on there. But the ozone concentrations in Redlands, the final ozone concentrations, looking at the lower panel here, were comparable, if not even a little bit higher than what we saw in Pasadena. And again, that's sort of maybe consistent with the fact that maybe Pasadena is a little bit NOx rich compared to Redlands. Now, what I'm showing here is the measured response to the perturbation in the final ozone concentration then. I've got 2 sites now. So I've got Pasadena on the left and Redlands on the right. And I've only got 4 months. And so I've got July, August, September, October shown on the lower axis of each plot. The red box and whisker plots are the weekdays, and the blue or teal box and whisker plots are the weekends. And you can see there is a difference. And then in the background of all that, I've got the seasonal cycle that we had measured in Sacramento in the year previous, just as sort of a measuring stick to see how we're doing. I want to focus on the upper panels to start with, because the lower panels are really just a mirror image of it, and as we saw in Sacramento. So let's focus just on the NOx perturbations to try to keep things simple. What we see in Pasadena is that there's not a strong seasonal cycle. We see that we're pretty much in the NOx-rich regime all the time in July, August, September, October. I mean, there's exceptions to that, but by and large, that's sort of the overall conclusion that you would draw from looking at those results. Whereas at Redlands, we do see that seasonal cycle a little bit stronger. We see starting out in NOx rich, going into more of a NOx limited regime, and then coming back to NOx rich as we go through the July, August, September, October. That is completely consistent with the picture that we got from Sacramento, combined with the Tripoli data and looking at the seasonal trends. And it's two things. It's your proximity to the downtown core of a major city, and then it's month of the year that seem to drive this. And that's completely consistent with what we see at these two sites in Pasadena and Redlands now. So again, we think that it's all making sense. It's hanging together in a very coherent way, and our interpretation of things seems to be consistent with all of that. Now, again, we're interested in what controls the ozone formation on the days with the highest ozone. Is it NOx limited or NOx rich? And so we wanted to try to plot things as a function of the ozone concentration, the MDA8 ozone. And so that's on the lower axis of each of these plots in Pasadena and Redlands. And then we've got the response to the NOx perturbation in the upper panel. Again, let's just focus on that, since the VOC panel down below is the mirror image of that. And what you see is that on the days with the highest ozone concentrations, sort of towards the right of each of those panels, you do see that you're moving more towards the NOx limited regime. You're moving sort of upwards relative to that dashed line at zero response to a NOx perturbation. So it's interesting, that happens at warmer temperatures. The colors of each of these box and whisker plots correspond to warmer temperatures, and that makes sense. But I think what's even more interesting is that even though the median response sort of heads towards almost the ridge line, you see days with extreme NOx limitation or extreme NOx sort of richness, VOC limitation at those high concentrations. The scatter in those box and whisker plots is going up as you go to the higher concentrations of ozone. And that's interesting. I mean, you can have a high ozone day that's either very NOx rich or very NOx limited. How do you design a control program for that exactly? Is picking one day really realistic anymore? Or do we need to start thinking about this statistically? What's the scatter in the data? How do you control 95% of the high ozone days? And that's what I'm going to try to get into sort of as we get into the modeling of all this to try to interpret the data. So really interesting trends. Now, we did model this with our tools that we have available to us. We use the SAPRAC 11 chemical mechanism. We normally drive that in a whole chemical transport model, but we have box models of all this. We built them to answer questions related to the Sacramento data set. And so we can apply that to the measurements here as well to try to understand things. What we see in the lower plots, if I just focus on those to start with, that's the predicted versus measured final ozone concentrations in our chambers. Really good agreement in that. But then if we look at the upper set of plots, I'm looking at the predicted versus measured perturbation response. So, how much does ozone change as I perturb the NOx concentrations at Pasadena and Redlands? And we're really good at that as well. So, the models work. The chemical mechanisms aren't missing something. We can reproduce the measured trends really well. Now, of course, this is helped tremendously by all of our collaborators who made great measurements during the field campaign. And so we have great VOC measurements to seed the box model calculations with. And that's part of the success of this exercise. But we're going to use this to try to understand then statistically, what does it all mean? So I'm creating isopleth plots now for Pasadena and Redlands in the upper and lower parts of this diagram. There's a lot going on here in addition to just the base isopleths. So I'm looking at isopleths under different ozone concentrations. So I'm going to create an isopleth for the low ozone concentrations of the left side of these figures. And then as I move right, the panels are creating isopleths for successively higher ozone concentrations. Ozone greater than 90 ppb if you look at the lower right plot. Or greater than between 70 and 90 at Pasadena, the furthest right you can go on the upper level. So Pasadena, again, didn't have anything over 90 in terms of final ozone concentrations in the chambers. Now, I'm showing the ridgeline here as the diagonal dashed line. That's all fine. That makes sense. These are all based on model calculations, the colors. The circles are measurements in the chambers at the same time, and they're largely consistent with all of the model predictions here. And so that, again, is a confidence building exercise that the models are working right. We can interpret these models accurately. But again, the scatter in the data is what I want to point out. Those dots are all over the place in these diagrams. There's not one day, there's not one sort of answer that it's always NOx rich or it's always NOx limited at those high ozone concentrations. There's a whole range of results. And so we thought about this, statistically then, you got to try to interpret this. And, based on the realization that the biogenics are a big part of the ozone formation, the VOCs that are driving ozone formation, we really wanted to focus on NOx control because we think that ultimately is what we're going to need to do to achieve compliance with the NAAQS. And so we wanted to know how much NOx control do we need to get down to NOx limited conditions if that's going to be the key to controlling ozone long term. Short term, there's certainly room for VOC control and NOx control combined, but long term, we probably need NOx control. And so we tried to look at what's the 95% confidence sort of interval for all of our measurements. And that's represented by the dark rectangles in each of these measurements, or each of these plots. And we wanted to calculate the distance between the upper right corner, the worst case scenario for the highest ozone concentrations in that sort of 95% confidence box, to the ridgeline. How far do we have to go in order to get the chemistry back into the NOx limited regime, where we think that further NOx controls will reduce ozone concentrations? And, there's a lot to take in these plots here, but what it boils down to is it takes about a 40% NOx control to get to the ridgeline. And further NOx controls after that will start to reduce ozone concentration. So that was a really useful statistical analysis of the modeling that's sort of matching these measurements. So, the summary of this recap field campaign, I'll just put up here, but, we had different trends in Pasadena versus Redlands because, again, one's closer to the urban core and one's further away. So we saw seasonal trends, or we didn't based on that. We have this big scatter in the data. Even at the high ozone concentrations, it might be NOx rich or it might be NOx limited. How do you design a control measure for that? We think that the models are working. We have confidence in that. And so we think that we can trust the model analysis then, that if we reduce the NOx concentrations by about 40%, we at least get to the ridgeline, and then you can start making progress on further ozone reductions. So that's a long way to go. We've done this same sort of analysis for more than just Redlands and Pasadena. We've done it for San Francisco, Sacramento, and Fresno. We didn't have the benefit of measurements there, so I can't put any measurement dots on these plots, but I can do that same calculation with the box, the rectangle showing the 95% confidence interval. And we can come up to an analysis of what it means for other cities across California. So I want to take a second here and just talk about COVID-19. That was one of our main questions that we asked. Why didn't we see ozone reductions during COVID-19? Traffic reduced by about 50% during the shutdown periods in April, March, April timeframe, and that reduced NOx concentrations by about 30%. We just talked about the fact that, you need about a 40% reduction to get to the ridgeline in Pasadena and Redlands. It turns out that it's not quite as strong in sort of smaller cities. You might not need quite that much in the summertime, in the middle of the summer. But in the springtime, when this shutdown happened for COVID-19, you're definitely NOx rich in all those locations. Think back to the Tripoli plots that I showed earlier. You're NOx rich everywhere in the summertime or in the springtime. And so the timing was just misaligned. If we would have had that 30% reduction in the summertime, I believe, based on these calculations, that you would have seen a slight reduction in ozone concentrations in small to medium-sized cities in California. But it happened in the springtime, so you didn't see that. In the urban cores of major cities, you can't get it with traffic alone. It's just not possible. You need to combine those reductions from traffic with other reductions in order to get to the NOx limited conditions. And we've done a lot of modeling with other studies to show that, but I won't go into the details here. So COVID-19 reflections, that was one of the two major questions that I wanted to address. And I think that we understand it now. So the final thing, what's the source of the VOC that's driving it? Again, we had these measurements during the recap campaign, very detailed VOC measurements some that we made, some that our collaborators made. And we wanted to try to interpret that to figure out which VOCs are driving ozone formation. Really, this is boiling down to three things. The biogenics that I noted earlier, which is kind of consistent with the seasonal profile, the volatile chemical products that our collaborators have noted are, must be a big chunk of things based on the ambient measurements. And then also traditional, motor vehicles, gasoline evaporation, and other things like more traditional issues in Southern California. We want to understand which of those three things is driving that ozone formation. So our measurements, again, as a part of this, we collected tube samples and sent them back to the lab at UC Davis. We measured a sort of the VOC composition with sorbent tubes every day where we made an ozone measurement. So that was a full, 120 days, 4 months that we were out in the field. We made these measurements to try to do this analysis. Just a quick picture of what's around the Redlands sampling site in the star here, this zooming in. We got the normal gas stations, there's dry cleaners, there's various educational institutions and things like that. We definitely saw trends when we got to the fall when, kids went back to school. When we switched over from summer blend gasoline to winter blend gasoline, like we saw things in the data set that were very sort of notable. We're going to analyze all of this with positive matrix factorization to start with, to try to find the factors that we think can be identified as different sources. And then we're going to then look at the ozone formation potential of the factors and try to estimate which are the ones that might be driving a lot of ozone formation. So, these PMF studies are complicated, and I'm not going to try to go into all the details. We probably have more material here than I can really explain fully in 1 hour. But we do have then, these trends and these factors that come out of the PMF analysis that we think we can identify. The biogenic one is there. The summer blend and winter blend gasoline are there. You can differentiate them by the butane content. They pull butane out of the summer blend gasoline because it's very volatile, and we want to have lower volatility during the summertime and put it back in the winter blend. You can see that clearly. And you can also see in the time trend of those sources, you see the winter blend gasoline signal jump up in the fall. You can see various, markers for educational institutions, the cafeteria starting up again, things happening at the schools around the site as we move into the fall when things start up again. So lots of different factors that we can resolve here. We also see our VCPs, the ethanol, the siloxanes, the classic markers for those volatile chemical products as well. So we think we can find all of these things in our data set. We got names that we think make sense, and I won't go into all the rationale for that, but just to say that this is based on the chemical signature and the timing and the wind direction when we tend to see those things all coming together to try to, apply the art of naming factors in PMF. This is not a science. There is some art in this. And so these things, I think, are subject to reasonable discussion and interpretation. And happy to have a more in-depth conversation about that if anybody wants to. So if we take the ozone formation potential that we've measured for all these different compounds and look at the factors that we found and sort of take a weighted average of all those different VOC concentrations times their sort of incremental ozone formation, what we find is the following pie chart of VOC contributions to ozone. I want to start in the lower left because, biogenics has been a big part. We think, based on this analysis, that biogenics VOCs drive about 23, 24% of the ozone formation at Redlands across that 4-month period. The VCPs, the ethanol factor and the siloxane factor, account for on the order of 20%. So just slightly less than the biogenics. So still significant, but not dominant. We had a big, what we call plant decomposition that was characterized by methanol, very high methanol spikes. That's not VCPs. Methanol is not something that typically is a part of that necessarily. So we called that plant decomposition in our analysis, but again, subject to interpretation perhaps. And then we have, summer blend and winter blend gasoline combining for, in the lower sort of part of this pie chart, around 12, 13% of the sort of contribution to ozone formation. Now, we were a little worried that this is a simplistic analysis that really the VOC sort of forming the ozone might be influenced by the NOx sources that happen to be sort of nearby or in the same sort of wind trajectory. So, we might be somehow biasing things by this sort of simplistic interpretation of just multiplying the composition profile by its incremental reactivity. So we took again our box model results that we thought were pretty accurate in predicting the final ozone concentration or the perturbation response to ozone. And we analyzed the ozone formation potential. We have a tagged mechanism within our box model. And so if we divide the sources up across those different factors that I said, and then run it through the calculation, I don't have to just multiply by a linear incremental reactivity response factor. I can actually watch it form ozone based on the full SAPRAC 11 chemical mechanism. And with tagging, I can say then what is the ozone formation from each of those sources. When you do that, you come up with this picture, and it's very consistent with what we saw before, with the biogenics being about 24%, the VCPs being about 21%, the winter blend and summer blend gasoline, being somewhere 12, 13%. And then, you've got the other sources contributing. So we think that this all hangs together. One thing that is noteworthy is that around 40% of the ozone couldn't be apportioned using this technique. We can only apportion the stuff that was, going to form from VOCs on that day. And so the other 40% formed yesterday or the day before. We think that maybe this sort of source split for the 60% that we do know about is our best guess for the 40% that we're sort of blind to with this technique. But really what we need to do is use chemical transport models that can go backwards multiple days in order to interpret this. So in summary for this part of the study, we found 9 factors with the PMF analysis for contributing to VOCs at Redlands. And the major ones that contributed to the ozone formation, that plant decomposition, biogenic, the VCPs are the top 3 sources. We also have the contributions from the traditional mobile sources, the gasoline-powered vehicles. And that we think that with these sort of techniques, we can explicitly analyze 60% of the ozone formation that happens on that day. And you have to use chemical transport models to go further backwards in time. So that wraps up everything that I wanted to talk about today. I've gone almost an hour. I promised Sheng I would cut it to 50 minutes, but I went a little bit long. But I do want to finish with acknowledgments. So, of course, we have the funding from the CARB project, but this was matched up with complementary funding from a number of different sponsors, and support. So we have CRC helped us with the VOC analysis that I just talked about. The UC Davis Institute for Transportation Studies funded the construction of the second set of chambers. The UC Davis Air Quality Research Center, provided instrumentation and, gave us another trailer to bring the chambers out into the field with. The South Coast Air Quality Management District hosting us at Redlands. And then the Caltech and the NOAA teams during recap, of course, this was a huge team effort. And then there was a big team of people behind all of that as well that I should acknowledge, both people at UC Davis, at CARB, at South Coast, at Caltech, at NOAA. It's just a huge effort involved in this. And as I said, this thing snowballed because it was so exciting. At the end of the day, I think we were really excited about everything that was coming out of it, and that momentum built. And so this was in my perspective, a great project, and I really enjoyed working on it. So with that, I'll finish up, and happy to discuss with any folks that are still online.
Thank you, Mike. This is a very nice presentation. And now we enter the Q&A session. First,
I'll leave my slides up for right now, just in case someone wants to refer back to them. So I'll leave it up for right now.
All right. Any questions?
I see 5 in the chat.
I see one in the chat. So Daisy is asking, is the study available for access?
So yes. So we will post all of this data once the publications are accepted. And so it is our intention to make all of this data publicly available up on a website that either we will host or CARB will host or both. And there is, I think, is a recap field site as well. So the answer is yes. We're going to make all of this public, and happy to share this and discuss further with anybody that's interested.
Thank you, Mike. I just want to add that the final report for this project will also be posted on the CARB's webpage that I sent earlier in the chat. All right. Next question. There's a question about siloxane. Did you track D4 and D5
Yeah,
siloxanes for VCPs?
yeah, both were measured. I can't remember which one specifically, or whether both of them were in uh, the factor, but let's take a look. I'll go back upwards. This is one of these things that we scooted by really quickly, but it should be in here. I can't see it. Yeah, I'll have to answer that one offline. But I know that we tried to measure a broad suite of siloxanes. Oh, there it is, D5. It's the last one, the one at the very end there. So the D5 siloxane apparently is the one that we used in the factor.
All right. Next question is from Jason. He's saying, we know that over urban areas, the PBL is often not well mixed, and that NOx often has a steeper vertical gradient than VOCs. Is there any way to assess whether the measurements you made at the surface are representative of ozone production integrated over the PBL depth?
Again, I'm going to rely on all the corroborating evidence, especially with tropomi. In terms of understanding the column average versus the surface measurements, and so the timing of the filling of the chamber, I think, is really critical for this. Do you fill in 6 o'clock in the morning before the atmosphere really starts to mix, sort of before sunrise, or do you fill sort of after the atmosphere starts to mix? And so we were very intentional about that 10 to 12 time period to be early enough in the day to sort of capture initial conditions, but then late enough in the day where mixing was starting to happen. So I don't think there's a perfect answer to this question. I don't think there's one right time, but we did give it some thought, and we are encouraged about the level of agreement between the tropomi analysis, that satellite-based column measurement, and the ground-based measurement. I don't think we would claim that our method can ever sort of fully address this question. I think you do need to sort of rely on models at some point that capture all these processes at the same time. So I guess no way to completely be sure, but very encouraged by the level of agreement between different techniques that our measurements are meaningful.
All right, thanks. Next question is, how did you derive tropomi without fire information? And regarding to the urban study, should we need to consider the regional chemical background change by season?
So I want to go back to the tropomi picture where we had some of the fires pointed out. So let me try to back up a little bit. Yeah, so these are, in these pictures, these are fire boundaries that you see outlined here during 2020. So you can see in August, September, we did have some major fires going, and those are based on, reported fire boundaries. I don't know if we can fully trust the tropomi measurements inside those fire boundaries, again, because the optical properties changing so much. I'm not sure how the measurements that tropomi make would be influenced by thick, heavy, dense smoke plumes. In terms of the tropomi sort of interpretation of fire versus not fire days at the measurement site, which is the small triangle in the center of California here, we worked with our collaborators again at CARB, and I think Annie was an intern, wasn't she, at the time, or doing an internship? But someone did an analysis to sort of interpret the change in the PM concentrations day to day as an indicator of whether or not we had fire influence or not at the monitoring site. And that allowed us to then try to tag those days and maybe look them separately from the other days. So that was how we did it, basically. I'm not sure if a paper has been published on that technique yet or not, but I know there was one in preparation at one point to describe how that was done. So I think that maybe answers part of the question, but then what's the rest of it, Sean? Did I answer all
It's
of it?
about the regional chemical background change by season. I think this is related to your seasonal ozone sensitivity analysis.
Yeah. So I think the air that blows into cities passes over rural regions. I mean, definitely. So I think that those rural regions are, influencing what's happening in the cities. It's not just the biogenic emissions from within the city, but it's in the surrounding area that's happening as well. Now, if you're asking about what's happening based on air blowing sort of from the Pacific Ocean into California, we haven't thought about that as much. And I don't have as much sort of comment ready on that one. So if by background you mean what blows in from the Pacific Ocean, I guess I don't know. If by background you mean the sort of surrounding rural area around our cities, I think absolutely yes. We need to think about that as influencing what's happening in the cities.
Thank you. The next is a comment from Daisy. We were studying energy usage versus VMT before and after COVID. And in 2020, when highway VMT was lowest, energy usage increased. So it follows the exhaust emissions, including GHG. So interested to see your results.
I think that's more of a comment than a question.
Yeah.
So I'll
Right.
just say thank you. Yeah. As I said, this study snowballed because, it just got so interesting. So it tremendously successful from my perspective.
Mhm. The next one is also from Jason. Was the Tropomi cutoff of 4.6 derived just from Sacramento data or a mix of Sacramento plus South Coast Air Basin data?
I remember it being the Sacramento data set that we used for that. But I think Xing Lun's on the line, and so he can maybe answer that if we can unmute him really quickly, or if
Mhm.
not, then I'm sure he can answer that as a follow-up email. But I remember the 4.6 came out of the Sacramento analysis.
Yeah. The 4.6 is all based on the Sacramento measurement in 2020.
Yeah. Thanks, Xing Lun.
Thanks, Shanlin. Another question. This is a very conceptual question, comment. Our SIP attainment strategy must be demonstrated through our modeling. How could your findings help inform or further refine our inventory and modeling representation of what you captured?
Yeah, I mean, that's a really interesting
Overall.
question. And it goes back to what I was saying at the end with the scatter in the data. During those high ozone days, you're not just seeing consistent NOx limitation conditions or NOx rich conditions. You're seeing this range of conditions. And maybe I can bring that up again, flip down to that box and whisker plot for the modeling. Maybe it was back here.
Mhm.
Yeah, this is it. So there's not one answer. There's not one type of day in the high NOx or the high ozone concentration conditions, and so that to me really says that we have to start thinking about this in a statistical sense. How do we design a control strategy that would work on 95% of the high ozone days? And that's what we tried to do in the simple box and whisker type analysis that we, or I'm sorry, the simple box model analysis that we tried to do. But really, I think we need to start thinking about that in full chemical CTM, like chemical transport model analysis. Also, it's probably not sufficient to model a single day anymore. It's probably necessary to model a range of days, in my opinion. And that's definitely a change of perspective. Not just one design day. You got to think about 95% confidence intervals and those sorts of things.
Thanks, Mike. Any other questions or comments? Oh, there's another one. So there's evidence in the literature that the ratio of formaldehyde to NO2 cutoff vary regionally by a large amount. So please be cautious applying your 4.6 ratio statewide, meaning that it could be different in other regions.
Yeah, and that's well taken. I think when we published that number, we also looked at those other studies, and the 4.6 seems to fall into the range. But there is a range, and so we fully acknowledge that. And really, this is all just weight of evidence. That it wasn't a Tropomi study. It was a ground-based chamber study. We think that it all hangs together, and we think that the observations that we made in Pasadena, being sort of consistently NOx rich, are consistent with our interpretation of that 4.6 in the Tropomi data and sort of these more urban cores always being sort of NOx rich during the summertime even. So I don't think it's changing any conclusions of our or interpretation of our results, but I do agree that there probably isn't one number 4.6 that characterizes everywhere. So well taken.
Thanks, Mike. Other questions or comments? I don't see any. If not, I'd like to conclude this meeting by thank you again, Mike, for the very informative presentation. And thank you everybody for attending the meeting. Wishing all have a nice day. Bye.
Thanks, everyone.